So you've probably seen Punnett squares before. I'm going to try to present it in maybe a different way. A Punnett square is no different than just thinking through sexual reproduction. But we do some sort of shorthand, meaning like abbreviations, because we don't want to write out the name of all the genes and such. And so one thing we want to think about is, first of all, that Mendel never made a Punnett square, which if you hate Punnett squares, you might enjoy that fact. Mendel never used a Punnett square. In fact, a Punnett square is named after Reginald Punnett, and Reginald Punnett would have been 9 years old when Mendel died. So I don't think their paths ever crossed in real life. Also, Mendel lived in Eastern Europe and Reginald Punnett was British. So, but what a Punnett square is just an organized way of thinking through sexual reproduction. So I know this picture here is how it's usually shown. I'm going to start with a blank slate here. And I'm going to tell you what's being indicated when they show you a Punnett square. First of all, genes are on chromosomes. And so if you draw a cell, remember we said, okay, there's a homologous pair of long chromosomes and a homologous pair of medium chromosomes. Why did it do that? Hang on, I need to get rid of that. I don't like that. There's the homologous pair of long chromosomes. And then here's the medium chromosomes, maybe 2 of those. I'm showing diodes. And then the small chromosomes. Okay, whatever. So two of each chromosome, I'm showing the dyad form. But let's say on one of these chromosomes, there's the purple color allele. I'm going to put it as a big P. And on the other one, there's also a purple allele, big P. Now, I could write out the word purple, but the big P is kind of the lazy man's way of doing it. And when I do a capital letter, I'm telling you that I already know that it's the dominant allele. So, you know, after Mendel did the first cross and he figured out that purple was dominant, after that we know it's the dominant allele. So when I use the capital letter, I'm telling you that information. So big P just stands for the purple allele, and they're telling you that it's a dominant allele. Now, this would be like plant, this would be the P1 plant. Remember the grandparents in my story earlier. And the other grandparent had 2 big chromosomes, 2 medium chromosomes. I'm really getting sloppy. 2 small chromosomes. But on that homologous pair, that white one had 2 little p's, 1 on each of the homologous chromosomes. And the little p is going to stand for the white allele. Again, I don't want to write out the word white every time. I don't want to actually even draw the chromosomes every time. And so what happens is, instead of drawing out and saying, okay, the purple flower plant has cells that look like this, and the white flower plant has cells that look like this, what we do is we just write the genes we care about. I only care about the gene for flower color, and I only care about the 2 alleles. So big P represents this whole plant. And then I'm going to cross that plant. I'm going to breed that plant with this plant. But the only part of this that I care about is the little p, because that's what I'm looking at. I'm looking at flower color inheritance. Now, all the other chromosomes are there. We're ignoring them. And so this is the cross. This is the P1 cross, right? The true breeding cross fertilization. So I'm going to make some more room here. I'm going to just move this to the top, okay? So when we write this, big P, cross little p, what we mean is a whole plant, and in every cell of that plant, both copies of the flower color gene are purple, and we're going to breed that to a whole plant. In every cell in that plant, both copies of the flower color gene are white. Now, this plant here let's say this is the plant that's going to make the sperm. So when cells that have two big P's go through meiosis, each sperm was only going to get one P, one copy of that homologous chromosome pair. And no matter how you shake it, it's going to have to be a big P. So all the sperm have a big P. They all have a copy of every chromosome, and the big one has a big P allele on it. And when let's say this plant is going to be the plant that makes the eggs, all the eggs are going to have to carry a little p. In other words, you can't pass something to your children unless you also have it. And in this case, there's really no options other than big P from this purple parent, little p from this white parent. And so this will be the sperm, and these will be the eggs. I'm only drawing one, but they're all like this, and I'm only drawing one egg. And I always put a tail on my sperm so you can remember it's biological. So the chromosomes in this sperm, one of them has a copy of the purple color allele, and on one of the chromosomes in this egg, there's a white color allele. So when egg and sperm meet, that's called fertilization, and that's going to create a zygote and a whole organism that has in every cell a big chromosome with a purple allele and another big chromosome with a white allele. That's the homologous pair in that hybrid offspring, which is this here. So this is the F1 individual. And there's probably not just one offspring, there's probably hundreds, but they're all going to be big P, little p in their genetic makeup in their cells. So that's that first part of the monohybrid cross. But really this part just creates the hybrid. Then, remember, Mendel self-fertilized the F1 plants. And I need more space. So I'm going to move the F1 plant up here. And the F1 plant had a big P on one big chromosome and a little p on the other big chromosome. And so when this plant is going to make eggs and sperm, because remember, self-fertilization means the same plant provides the eggs and the sperm. So when cells like this go through meiosis to make sperm, some sperm are going to carry a big P, tail, and some sperm will get a little p, and they'll be about equal amounts. So some big P sperm, some little p sperm. And when cells from this same plant make eggs, some eggs will carry a big P, and some eggs will carry a little p on their big chromosome. I've already stopped drawing the chromosomes even, because frankly, I'm not a good drawer. But the letters represent the alleles on chromosomes in cells, in organisms. And so it's all biological. Now, when egg and sperm meet, any sperm can meet up with any egg, potentially. And so, let's say this sperm, let me get a different color of ink. Let's say this sperm meets up with this egg. Then the zygote will be big P, little p. I'm going to write that in blue, the outcome. Big P, little p will be the zygote, the offspring. If this sperm meets up with this egg, then the offspring will be big P, big P. If this sperm, or one like it, of course it can't be the exact same one, but one just like it, meets up with this egg, then you'll get a little p zygote. And if this sperm meets up with this egg, or one just like it, it'll give you a big P, little p zygote. And so these are all the combinations. But what you can see here is it's getting kind of messy. And this is only one gene with two alleles. Pretty quickly, Mendel starts to deal with 2 genes with 2 alleles each, 3 genes with 2 alleles each, 4 genes with 2 alleles each. You can see how pretty quickly it's going to be too hard to figure out all the possible fertilizations with this, I guess you could call it, line method. So that's where the Punnett square comes in. Just to kind of organize it, you put all the egg possibilities down the side, all the sperm possibilities up top, And then when you bring them together in one square of the grid, you're recreating a possible fertilization. So what we end up doing is, I'm going to rewrite this with a Punnett square. What we end up doing is we say, okay, and I usually still draw the circle so I can remember that this is biological. Some of the eggs carry a big P, some of the eggs carried a little p, some of the sperm carried a big P, some of the sperm carried a little p, and I put tails on my sperm. And these are outside the grid. And then inside the grid are all the possible ways they can come together. And I'm just keeping it organized so I know that I got them all. So big P and big P end up together, that's a zygote. Big P and little p end up together, that's a zygote. Big P, little p, that's a zygote. Little p, that's a zygote. Now, I'm not saying there's only 4 offspring, but when you reduce it down to that simplest ratio, it would fall into this pattern. For every 3 offspring that are purple, because remember, if you have just one big P allele, that's going to dominate and make the flowers purple. For every 3 plants that have purple flowers, 3/4 of the plants will be purple flowers, 1/4, 1 out of 4. I'm counting how many squares there are here. There are 4 squares. 1 out of 4 will be white. Little p is the only combination that gives you a white flower plant. But what's inside the grid has to be zygotes. So you just kind of have to figure out the gametes first. I don't use a grid for that. And then you put them across the top and down the side. Those are gametes. And then what's inside the boxes are the zygotes, the product of fertilization. All right, I'm going to take a break and move on to the dihybrid cross in the next section.